Hello everybody, thank you for your interest. My name is Richard Van and I'm excited to share the research that my colleagues and I had recently worked on, which compares the different data pre-processing techniques that can be applied on RNA-Seq datasets and how they affect predictions made by machine learning models across different test datasets. By way of outline, we'll begin with some background concepts. Then I'll state our big question, highlight some of the methods and results, and cap it off with conclusions and the key takeaway. Thanks to technological and biological advances in the last 2 decades, we can generate RNA-Seq data that provides a snapshot of all the expressed transcripts and their frequencies in a population of cells at a given time. We can use this molecular data to train a machine learning model to make predictions on unseen test samples. In this figure, the trained data will infer what labels, such as the tissue or cancer type, that is associated with each of these test samples. In our simplified example, we have 3 labels corresponding to the liver, gastrointestinal tract, and the lung. We first train the model with the gene expression data and known labels of each training sample. Then when we test the model, only the gene expression data of the test set is provided to the model and the test labels are predicted. One of the common challenges of machine learning classification is the data itself. A model will not be able to perform optimally if the data has systematic variation caused by random noise. An example of systematic variation is RNA-Seq data that comes from different sequencing centers. It would be ideal for the data to be from the same sequencing center so that the systematic variation is small. However, this isn't the case when you have thousands of samples collected at different locations, so more than one sequencing center is necessary. Trying to reduce this systematic variation in the data is the focus of our research. There are 3 broad types of data preprocessing methods to address this issue. The first one listed here is normalization, which is the transformation of each sample's data to be more comparable, such as making their distributions be statistically identical. Next, there's batch effect correction, which is the procedure to remove variability from data sets due to known batches, which in our previous slide, the batch variable would be the sequencing center associated with each sample. Then we also have data scaling, which is the process of transforming the values of the data set to be within a specified range. Values in a similar range allow more of an equal contribution to the impact on the model performance. With that being said, this brings us to our big question, that is, can these data preprocessing methods reduce the systematic variation found in our RNA-Seq data sets and thus help us build better performing models? Okay, let's take a look at the methods section of how we conducted this research. Here is an overall flowchart showing the data sets and the possible combinations of classification models that can be built from data preprocessing methods. We have 16 possible classification models based on the 4 types of normalization, whether or not batch effect correction is applied, and whether or not data scaling is applied. We then determine the performance metrics for each of the 16 constructed classifiers to evaluate them and compare them. Okay, let's take a look at our main results. In this experimental setup, we train with TCGA and test with GTEx data. We saw that half of our data preprocessing combinations showed an improvement in overall performance, basically any combination that had batch effect correction applied. Now I want to zoom in on the data preprocessing combination that provided the most improvement from the baseline. You saw this image earlier with the different sequencing centers. Here it is again in more detail. We can see that the GI or gastrointestinal samples for TCGA were all sequenced in British Columbia, while the non-GI samples were sequenced at the University of North Carolina. If we have the batch information for each sample, we can perform batch effect correction to remove this systematic variation. Here are the confusion matrices of the baseline unmodified data set on the left, and on the right we have the best performing modified data set, which has undergone batch effect correction, quantile normalization, and data scaling. You can see with the green circle here that the GI samples of the best performing modified data set led to around 500 correct predictions as compared to only 13 correct predictions in the baseline. So far, I've only shown you the positive result of our research, but this was not the case for all test data sets. We had a negative result here when we switched out the test data set from GTEx to ICGC Geo for this experimental setup. You can see here that no combinations of data preprocessing helped. In fact, most combinations led to a deterioration of overall performance. Alright, on to the conclusions. We showed that the data preprocessing methods were beneficial and improved classifier performance in the case where GTEx served as the test data set. The results suggest that batch effect correction was the most important data preprocessing method in this setup. And we also observed that data preprocessing approaches were detrimental to the classification task when the external test data set was from ICGC Geo. With that, a key takeaway from this research is the application of data preprocessing techniques to RNA-Seq data for classification of tissue types are not always appropriate. Here is my acknowledgement slide. I want to thank my PI, Dr. Mira Han, and my fellow UNLV Han Lab members. Also, my thanks to the Student Council of the International Society for Computational Biology for hosting this conference. And finally, I would like to thank you, the audience, for listening to my presentation. Thank you.